This section of lectures will cover the digestive system. If you are interested in seeing images of different diseases of the digestive system, there are several endoscopy atlases online with great pictures. Our body is set up as a tube within a tube. The digestive system is the inside tube. Our skin makes up the outside tube, and the rest of our body is in between. This is easier to visualize in a snake, where the digestive organs are all in a long line, where ours fold up in the abdominal cavity. There are high numbers of bacteria in our digestive system. These are essential for the digestive process. There are more bacterial cells in your gut than human cells in your entire body. The tube is segmented, with each segment forming the next organ in the line. Our digestive process is a combination of both mechanical and chemical processes to break down the food. Digestion begins in the mouth with chewing or mastication. Along with chewing, we combine the food with saliva to form a bolus. Saliva contains salivary amylase to begin breaking down starches in the mouth. This can be seen if you set a small piece of bread or a cracker on your tongue. You can watch it begin to break down as it sits there. There is also a small amount of lipase present in the saliva to help remove the fatty residue left in the mouth after eating fats. In general, enzyme names will end with ASE. The tongue is used to taste foods, but it also helps to hold food in place. Humans have heterodentition since we have a variety of types of teeth. The carnassials are the front teeth and are designed for scraping. The incisors are sometimes called canine teeth and are the pointy teeth in the front side corners of the mouth that are designed for piercing and tearing. Compared to animals with a predominantly carnivorous diet, such as a dog or a wolf, we have very small carnassials. The molars are the large flat teeth in the back. These are meant for grinding and are seen throughout the mouth of an herbivore animal, such as a cow. This variety in teeth suggests the need for a variety of types of food in the diet, but not a high-meat diet in particular. Fats are also warmed in the mouth to help with their breakdown later on. The esophagus is the tube that connects the mouth to the stomach. The upper portion of the esophagus is under voluntary control, but the lower portion is involuntary. You have noticed this if you swallow something too large or too hot. You cannot choose to make it go down any faster. The epiglottis is the elastic flap that covers the trachea. This keeps food from going down the airway. The action of the epiglottis is poorly coordinated in babies and in the elderly, causing a greater risk of choking. Peristalsis is the rhythmic wave-like muscle contraction used to propel food through the digestive system. This action can easily be seen in a snake when they swallow an animal like a rabbit. The same squeezing process occurs in humans, only we take smaller bites and have more cushioning around our digestive system to hide the activity. This illustration shows the gastrointestinal tract. It gives a nice picture of the relative locations of each of the organs without being cluttered up with too many other things in the picture. The stomach is a stretchy sac where the next steps of digestion occur. The cardia is the top portion, the pylorus is the bottom portion, and the fundus is the belly. The food is called chyme after it is digested in the stomach. Gastric rugae are the folds on the inside surface of the stomach. These help to allow the stomach to stretch as well as increase the surface area. The outer portion of the stomach is made of 3 layers of muscle that run diagonal, longitudinal, and circular in direction. This allows stretch and contraction of the stomach during and after meals. The cardiac valve is at the top of the stomach and prevents the contents from going back up into the esophagus. Acid reflux occurs when the acidic contents move back up into the esophagus, which is not designed to deal with the acidity. The pyloric valve is at the distal end of the stomach and prevents the contents from passing into the small intestine until they are ready. This is triggered by a pH change in the contents. Goblet cells secrete mucus to coat the inside of the stomach and protect it from the acid. Hydrochloric acid is secreted by parietal cells in the stomach. It is used to activate the pepsinogen to pepsin. Pepsin is produced in the inactive form as pepsinogen from the chief cells and is an enzyme to break down proteins. The only nutrient broken down in the stomach is protein. There are no enzymes for dealing with the carbohydrates or fats. Most organisms would not survive the acidic environment in the stomach, but Helicobacter pylori is a bacteria found that is known to cause ulcer pain. This is not the only cause of ulcers, but it is a cause of many of them. This type of ulcer can be treated with antibiotics. Here we have a brief animation on gastric secretion from McGraw-Hill.
Regulation of stomach secretion is divided into 3 phases: cephalic. Regulation of stomach secretion is divided into 3 phases: cephalic, gastric, and intestinal. In the cephalic phase, the taste or smell of food, tactile sensation of food in the mouth, or thought of food, sends nervous impulses to the medulla oblongata. These impulses cause parasympathetic neurons via the vagus nerves to stimulate secretion of hydrochloric acid and pepsin in the stomach. The parasympathetic stimulation also results in secretion of gastrin from the lower part of the stomach. This hormone travels through the bloodstream and further stimulates hydrochloric acid and pepsin secretion in the upper and middle parts of the stomach. In the gastric phase of gastric secretion, food has entered and distended the stomach. This distension activates a parasympathetic reflex via the medulla oblongata and also has a direct stimulatory effect on the gastric glands. The result is continued secretion of hydrochloric acid and pepsin. In the intestinal phase of gastric secretion, chyme has entered the duodenum, so gastric secretion is no longer needed. When the chyme contains lipids from the digestion of fats or contains enough hydrochloric acid to bring its pH below 2, gastric secretion is inhibited. The lipids and hydrogen ions inhibit gastric secretion by 3 simultaneous actions. First, they cause impulses to go to the medulla oblongata to decrease parasympathetic stimulation of the gastric glands. Secondly, they set up local reflexes via neurons in the wall of the gut that decrease gastric secretion. Thirdly, they cause the release of 3 local hormones which travel via the circulation to the gastric glands and inhibit their secretion.
There are 3 parts to the small intestine: the duodenum, ileum, and jejunum. The large intestine is also known as the colon. Inside the small intestine is lined with finger-like projections called villi. These help to increase the surface area of the intestines, which is important since this allows absorption to take place. The villi contain lymphatic vessels called lacteals to absorb dietary fats, and the blood vessels to absorb other nutrients. The microvilli are additional cilia-like structures on the surface of the villi to even further increase the surface area. The small intestine also has goblet cells to produce mucus to help protect the lining from the acidic contents of the stomach it receives. The crypts will help produce some of the digestive juices used in the small intestine. Bacteria are extremely important here. We have numerous good, healthy bacteria living in our gut that are actually necessary. The exact mix of these bacteria in a person's gut largely depends on what they eat, but there is also variation in places in the world. Occasionally when taking antibiotics, we may disturb the balance of good bacteria in the gut and find it helpful to supplement our diet with more of these bacteria. If a person chooses to take these supplements, they would want to make sure they are purchasing refrigerated bacteria, as they will frequently have a high number of organisms killed by high temperatures and transport in the back of hot trailers. The gallbladder is a sac-like organ designed to hold bile until the appropriate time to release it into the system. Remember, the bile is made from recycled red blood cells who begin their recycling in the liver and spleen. Emulsification is the process that allows fats to combine with water. Soap acts like an emulsifying agent, especially when you have a greasy pan sitting in water and you add soap. You can watch the floating oil move away. The release of bile is triggered by cholecystokinin. The ampulla of Vater is the opening in the small intestine where the gallbladder connects to the small intestine and is involved in digesting food. The sphincter of body is the ring-shaped sphincter muscle used to close the valve at the ampulla of Vater. We have a quick animation here that's going to show the release of bile. So we can see where we have lipids, CCK, the cholecystokinin, and bile. So the lipids being released, triggering the cholecystokinin to release the bile and breaking the lipids down. Clusters of lipids pass from the stomach to the duodenum. The lipids cause the cells lining the duodenum to release CCK. CCK travels by the bloodstream to the gallbladder, and the gallbladder releases bile into the bile duct. Bile enters the duodenum and emulsifies the fats. It breaks the lipid clusters into single lipids so that lipase, an enzyme, can work better. The exocrine portion of the pancreas is active in the digestive system. It produces many enzymes to assist in the chemical breakdown of food. Some examples of these are maltase to break down maltose, sucrase to break down sucrose, lactase to break down lactose, peptidase to break down proteins, pancreatic lipase to break down lipids, ribonuclease and deoxyribonuclease to break down the genetic material, and bicarbonate to neutralize the acid from the stomach. The contents are delivered to the small intestine through the common bile duct shared with the gallbladder. The alpha cells secrete glucagon to increase blood glucose, while the beta cells secrete insulin to decrease blood glucose. We have an animation here of bicarbonate neutralizing the acid. When acid from the stomach enters the duodenum, cells lining the duodenum sense the acid and release the hormone secretin into the bloodstream. Secretin travels to the pancreas, where it stimulates the release of bicarbonate ions, which enter the duodenum and neutralize the acid. Lactose intolerance occurs in 70 to 80% of the human population. This is the inability to digest milk sugar or lactose because of lacking the enzyme lactase. Lactase levels are highest in infancy when a human consumes milk from its mother, and then begin to decrease. A genetic mutation occurred in some humans that allowed a group of them to continue digesting lactose into adulthood, but that is not the way we started out. When observing nature, no other animal consumes milk into adulthood, and no other animal consumes the milk of a different species. Although the dairy industry would have us believe we all need cow's milk, historically a human would not have been very likely to attempt to drink from the udder of a wild cow. While milk does contain a high amount of calcium, there are other sources of calcium with lower levels of sodium and phosphate. Harvard has actually recently released a study where they are no longer recommending dairy products in the diet as a necessity.